Hello everyone, my name is Debojyoti Chakraborty. I am from the CSIR Institute of Genomics and Integrative Biology in New Delhi, India. And today I'm going to talk to you about recent work where we have used CRISPR system through an affinity-based method as a diagnostic for resource-limited settings. We have been using it for multiple different applications for the detection of the SARS-CoV-2 infection, as well as for single nucleotide variants such as those found in diseases or such as those which are signatures of variants of concern. So I'll give you a little bit of background as to where we started from. Uh, my lab has been working extensively with Dr. Shobik Maiti in trying to characterize an orthogonal Cas system from Francisella novicida. This was a very naturally occurring, highly specific Cas9, which we reported in this paper in PNAS in 2019. It had a low editing efficiency inside cells. However, under in vitro conditions, what we did find out was that it had certain interesting properties due to which it was very, very specific in distinguishing two DNA substrates which were different by even a single mismatch. Why this protein has evolved differently, or what are the structural differences between other cases and this, is a matter of ongoing investigation. But what we clearly saw was that if you present it two substrates which are different in their sequence by, in this case, as you see in this agarose gel electrophoresis, by two nucleotides or three nucleotides, whereas the canonical Cas9 from Streptococcus pyogenes would be cleaving these off targets which are mismatched, the Francisella novicida Cas9 would not be cleaving them. We later on found out that not only was this Cas9 able to distinguish between the substrates on the basis of in vitro cleavage, but also they are not binding to these mismatched substrates, meaning that the specificity is also extendable at the level of interrogating with these substrates. Hence, the presentation is termed as affinity-based CRISPR diagnostics. This is what is the basis for the Feluda platform. The full form of Feluda is FN-Cas9 editor-linked uniform detection assay, and the name actually is an acronym which is also the name of a fictional detective character who is very commonly seen and is quite famous in Bengali literature, creation of Satyajit Ray. This particular technology was also licensed during the COVID times for detection of COVID non-exclusively, and a company called Tata Medical and Diagnostics came out with a product that was validated and approved in India for commercial use by the Drugs Controller General of India sometime in September last year. So how does Feluda actually work? As you will see that the principle of mismatch discrimination happens at the level of the Cas9 RNP interacting with the target DNA. So what we figured out is that if the guide RNA, which is very similar to the guide RNA as Streptococcus pyogenes Cas9 in terms of the way that the complex interacts with DNA, if this particular guide RNA has bulges due to mismatches, the FN-Cas9 protein is unable to interrogate with the DNA substrate anymore. And precisely, we can walk along the whole length of this guide and find out which are the points or which are the positions where this loss happens maximally. And this has been then taken into account for designing specific guide RNAs that can then distinguish two nucleotide sequences different by even a single mismatch. So therefore, if the Cas9 is an active form of Cas9 or a nuclease form of Cas9, it recognizes the DNA and then it cleaves the DNA, And such a readout can then be used either on an agarose gel electrophoresis, or it can also be used through a capillary electrophoresis technique. However, if we are using a nuclease deficient form of this Cas protein, which simply binds to the DNA but doesn't cleave it, then in that case, the affinity can be used to discriminate between a fully matched substrate versus a single mismatch. Later on, as I will show you, this can also be converted into a fluorescence-based or a paper strip-based assay of readout, very similar to that of a pregnancy test kit. Now, what we found out was that this high power of discrimination can be used to detect, of course, DNA as well as RNA if one involves in vitro transcription, uh, a step where the RNA is converted, a reverse transcription step into where the RNA is converted into DNA. It works on a stoichiometric scale where even the binding based assay can be converted into a method by which one can distinguish even zygosity. So heterozygous SNPs can also be detected, such as those seen in carriers of the sickle cell mutation. So in order to convert it into a fluorescent assay, we use a fluorescently labeled form of Cas9, and then by binding to the substrate, which would be biotin labeled, one can then detect it by a fluorescence detector. Or in the case of sickle cell for carrier screening, where the carrier screening is extremely important for preventing the growth of sickle cell disease, because it happens only in a homozygous state. So if you can do counseling at the level of carriers, then it is very useful. So we could also detect with 100% success in blinded samples both the homozygous as well as the sickle cell disease. During the same time, our colleagues from the All India Institute of Medical Sciences in New Delhi came up with this interesting proposition whether we can detect point mismatches in the Helicobacter pylori infection in patients, which causes peptic ulcers. Now, this Helicobacter pylori is generally can be treated with certain types of antibiotics, but in some special cases, when this particular bacteria has got point mutations in them, they become antibiotic resistant. And in such cases, these samples have to be then cultured, and after 3 to 4 days, one knows whether it is an antibiotic resistant form of the bacteria or not. Now that is not very useful because that takes a lot of time in the lab. Um, if we do a Feluda assay on this, which can detect this point mutations very efficiently, we can actually bring down this whole time to just an hour or 1.5 hours instead of this 3 to 4 days, and we could develop a technique or an assay also for Helicobacter pylori. Finally, we wanted to see whether this kind of an approach can work in the field. And as some of you might know, that the sickle cell anemia is a big health burden in India, as it is in several other countries. And unfortunately, most of the people who are affected by this disease come from tribal areas where the access to healthcare diagnosis is very poor. And normally in such places, the solubility test, where a very crude estimate of whether the person might be a sickle or a carrier, is done by taking blood samples from individuals and then looking at the precipitation. On the top left, we are seeing how the solubility test is performed, and on the top right, we are having a snapshot of a tertiary healthcare center, which is a primary school in this case, where children are lined up to see, to have their fingers pricked and the blood taken out. Some of them, like that little girl, as you can see over there, who doesn't have slippers on, is seems a little bit circumspective in trying to see what is happening there. And what we found out that we do not really need to use blood for Feluda. We can simply ask them to spit into a small tube, as is shown in the picture at the bottom, and then this tube actually contains the molecules that are necessary for breaking open the cells for the DNA to come out. and then the Feluda assay can actually be done on them very easily. So one can then detect whether this person is a sicker or a carrier. So we were basically trying to work on these principles to develop tests which can be used in the field. And this is when the COVID-19 pandemic actually struck. And we decided that if it can work well for single nucleotide polymorphisms or variants, we can actually also make it work for detecting the COVID-19 signature in a patient sample, where the most widely used technology is, of course, reverse transcription PCR. So to convert this into an assay for detecting the SARS-CoV infection, what we then did is that we developed a pipeline where the Feluda can be done on a paper strip. I will come to the technical details in the next slide how this works, but essentially a person's sample is taken either from the saliva or the nasopharyngeal swab, and then what we do is the RNA is extracted from these samples by breaking open the cells, and after the RNA is extracted, there is a single step a reverse transcription and PCR where the PCR reaction is carried out using biotin labeled primers. Now, because of the biotinylation in the primers, the amplicons that are generated have biotin labels on both sides. And these biotin labeled amplicons then serve as a substrate for the CRISPR complex to bind to and give a signal on a paper strip. I will explain the details on the next slide, but the major advantages of using this is that it has got almost three layers of specificity, where the reverse primer, the forward primer, and finally the high specificity, the Cas9, make sure that we can actually detect only the right amplicon. It is, of course, rapid. The entire assay can be done within an hour. It is economical because it doesn't require a real-time PCR instrument, but rather a normal PCR machine, or even an isothermal amplification where no machine is required. It is semi-quantitative as opposed to some the collateral cleavage-based assays, which uses different forms of Cas, and it doesn't require any specialized equipment like that of the real-time instrument. Now, how does this work on the paper strip? So what we have used here is the affinity of the Cas9 or the FN-Cas9 to bind to biotin labeled DNA. Now, this kind of a commercial paper strip has got gold nanoparticles right at the bottom, which are conjugated with antibodies which recognize the FAM motif or fluorescing amidite motif. And what we have done in the Cas9 RNP is that we have added a FAM label to the 3' end of the guide RNA. Thus, the RNP complex now has a FAM on one end. These gold nanoparticles can recognize this FAM. When we do the biotin labeled amplicon, if there is a positive amplification, which means that there is the patient is positive for COVID infection, then the Cas9 can recognize that this is a nucleus deficient format of Cas9. It can recognize that target and bind to it. Thus, the tertiary complex now has got FAM on one end, which is at the level of the guide RNA, and the biotin at the other end, which is at the level of the DNA amplicon to which it is bound. When this is recognized by the anti-FAM antibody conjugated gold nanoparticles in the strip, they together form another complex and they start moving up by lateral flow in the presence of a buffer. Now, as it moves up, it reaches an area which you will see marked in red, also called the test band. In this test band region, there is a high concentration of streptavidin. The streptavidin recognizes the biotin labeled complex and it gets deposited over there, forming a colored band. If the sample was negative and there was no presence of SARS-CoV signature, then the entire complex moves up and there is a control band which recognizes the anti-FAM antibody and therefore will give a band on the top. And this band should form regardless of whether the sample is positive or negative, and thus serves as a control for the assay. In order to make this work fine, we needed to develop the guide RNA in a chimeric fashion where the 3' can be FAM labeled and it can be then formed in a chimeric one along with the Cas9. And we needed to find out what would be the degree of overlap in the case of the sgRNA because this has never been tested for Cas9. We found out the exact conditions for this to happen, which would allow the Cas9 complex to form and successfully recognize the target, as you can see over here, showing the overlap of the tracer RNA cRNA, which is best suited at 19 nucleotides of overlap. And then using that, we could develop this assay where a typical assay would have this kind of strips with two bands. The bottom band is called the test band, telling you that a sample is positive. The upper band is the control band, which will form in every strip. So using this principle, we adapted this for SARS-CoV. We went through the entire genome of SARS-CoV, found out regions which are conserved and not present in other viral or human sequences, figured out what would be the right concentrations of temperature as well as primer concentrations, and also found out that this particular assay was highly sensitive to just a few copies of the target RNA in the initial sample. However, since we are looking at an image of a strip, or we are looking at a strip, we wondered if it might be possible to convert this into an automated system. So to do that, of detection basically. So to do that, we developed an app called Topshe, or True Outcome Predicted by a Strip Evaluation, which can be used to simply take a picture of the strip. And this app has been trained by machine language, where over several different types of data to come out with a prediction whether the sample is positive or negative, thereby reducing any kind of operator bias in what one perceives as a positive signal or a negative signal. We also developed a web tool called Jatayu, Junction for the Analysis and Target Design of your Feluda experiment, where the user can incorporate his target sequence, and the Jatayu returns the right primer sequences that would be necessary to perform a Feluda experiment. So this is to support the performance of a Feluda assay. We also made further improvements where we could convert this whole thing into just about half an hour or 45 minutes using isothermal PCR, using recombinant polymerase amplification, or RPA, where even without the need for any kind of PCR machine, just on a heating bath or a heating block or a water bath, one could perform this even in a one-pot reaction where all the components will be present together in the tube. And this could also be used to further reduce the time as well as the complexity associated with diagnosis. This, of course, went into the commercial spectrum, and currently this is known as Tata MD Check by the company which took this up, which uses this, and it has been also featured as a technology, and these are the people who have been principally involved in developing this technology. This has now also, by the manufacturers, been converted into a more mobile kind of a platform where it is being used in different parts of India at different destinations and different settings. And the methodology can be adapted essentially to a large number of different detection systems, which we are currently, or let's say detection outputs for different diseases, which we are also currently exploring. Now, what happened during the pandemic was that, of course, the SARS-CoV started mutating and adapting to the different selection pressures. And therefore, what we had was variants. These are the different variants which had created havoc across the world. I think several of us are aware of the Delta variant, which has been kind of the most deadly. And these are mutations that are normally arising in viral sequences as the virus moves from one host to the other. We figured out that normally these variants would be detected majorly through high throughput sequencing studies, and that is what is also being done across the world. Now, one can do that, but there are some disadvantages when one does these kind of high throughput DNA sequencing for diagnosis is that this is typically a high cost experiment, and it takes some hours to actually get the result. Typically, this can be anywhere between 2 to 3 days. A large number of samples needs to be pooled in order to save on manpower and cost, and it is also only able to have a very how to say robust calling mechanism for mutations when the samples are very concentrated, which means that high CT values are often missed due to the chemistry of the sequencing methodology. On the other hand, RT-PCR, which is relatively quick and the method of choice for normal detection of COVID, sometimes can not work so well for point mutation because the Tachman probe chemistry, which works on binding to the target, has to be extensively tested for mismatches, and it is not that efficient for doing this kind of mismatch-based discrimination. So what we decided to come up with is a platform where the CRISPR system, due to its high affinity with full matches and very low affinity with mismatches, can be adapted to a platform called a RAY, or rapid variant assay, in which one could distinguish these variants of concern as well on a paper strip. So in order to do that, we again took the advantage that with a single mismatch or a complete match, the Cas9 binds to the substrate, whereas with a couple of mismatches, the Cas9 is evicting itself from the substrate. And it is relatively universal in terms of discrimination across multiple targets and multiple different substrates that we have tested the positioning of these mismatches. Thereby, we can actually adapt this very easily for different types of targets. And some of the targets that we specifically chose for these applications were mutational signatures of these variants of concern. As the virus mutated, it developed new kinds of signatures, and we tested this methodology on the alpha variant first, which was the N501Y mutation. As you can see, this variation appears in the S gene of the virus, and if you look at the sequence shown over here on in the slide, there is a nucleotide which is A highlighted in yellow in the sequence, which is the variant form of the or let's say a signature of the variant form of the virus, which is the alpha variant. And by designing a guide which would specifically be able to target this, and another guide which would be able to target the virus in itself, we could use the combination of these strips, two strips to be exact, to come up with a signal or an assay where if the guide is binding to both the N501Y as well as an other region which doesn't get mutated, then both the strips would be positive, telling that this particular sample is an alpha variant. On the other hand, if the guide binds only to the wild type but doesn't bind to the N501Y mutant, then one knows that this sample is a COVID affected positive sample, but it is not positive for the alpha variant. And this is what you see on the bottom in terms of the reproducibility of the assay over different samples, that it always is able to distinguish between what is called the N501Y positive versus negative samples. We of course used a large amplicon over here, and we found that there were some problems if you use larger amplicons in a PCR reaction, because sometimes the amplification is not very faithful, and it also takes a longer time for the PCR reaction to complete. So what we went to was a double amplicon ray, which were very short amplicons that we made, and then the readout can be done on a paper strip within a period of about 55 minutes. So you can imagine that this is several fold orders faster in magnitude than if you would do a sequencing reaction. We also came up with ideas to figure out what would be the right concentrations, where to put the biotin labels in these amplicons, and then we can see that up till a CT value of close to 35, we can specifically distinguish between a substrate which is SARS-CoV positive and also alpha variant negative versus a substrate which is both positive for infection as well as is a variant of concern because of the presence of the mutation. Interestingly, this particular technology of variant detection can actually be extended to cover the majority of variants of concern, because, like I said, the mutations that are detected and the presence of the mismatches where it should be there on the guide RNA can actually is quite universal. So this means that the mutational signatures of all the variants of concern can be captured using this array platform. And the SN-Cas9, due to the way that it recognizes the DNA, can give the two different types of signal, where one it is bound to the substrate strongly, and in the other case it is evicted from the substrate. Thus, this is a platform that is not only very useful for the variant of concern for SARS-CoV diagnostics, but it is also useful if one wants to use it for any kind of other single nucleotide variants, such as those found in diseases, or those associated with certain types of traits, or those which are just present in the DNA and one needs to detect them for a particular assay. Uh, these kind of assays can help in multiple ways. One of the ways that it can help is for early detection of variants, and this is something that we had, we had uh, uh, put forward in a Lancet commentary this year, where the variant diagnosis, which normally takes place by high throughput sequencing, can actually be streamlined by having initial cutoff based on a rapid diagnostic platform such as through CRISPR. One can detect these variant signatures and then can very nicely uh, decide on the fate of such individuals, whether they need to be quarantined in a stronger way or they need to be quarantined in a normal, home quarantine, or they can be released. Because certain variants are more susceptible for transmission, as has been shown earlier, than the others. So these are the type of screening approaches that one might potentially benefit from if one has diagnostic assays which are so specific for detecting these point mutations. So with that, I come actually to the most important slide, which is the acknowledgement slide. And there are a large number of people who have been working on this for a long period of time, almost 1.5 years now. During the entire pandemic, these are students, postdoc scientists, lab technicians, and we are a big team who have been working on this for a long period of time actually, and we have been generally funded very generously by different funding agencies, both government and non-governmental. And I'm extremely glad to get an opportunity today to present this before you, and would be happy to address questions if you might have based on this. Thank you very much.